This explainer will analyze the paper by Huen and colleagues titled A Spatially Resolved Atlas of the Developing Human Cortex. This study constructs a spatial transcriptomic atlas of the human fetal cortex to address a fundamental limitation in developmental neurobiology. For decades, our ability to classify cell types has far outpaced our ability to map them within intact tissue. Conventional dissociative single-cell methods provide a comprehensive parts list of the brain, but in the process, they destroy the very spatial information that's necessary to understand how those parts assemble into functional structures, like cortical layers and areas. The central argument of this paper is that by preserving the spatial context of individual cells, it's possible to uncover otherwise invisible principles of cortical development. Specifically, the authors demonstrate that the 6-layered neocortical structure is specified molecularly months before it's visible morphologically, and that cortical areas are established through 2 distinct modes of spatial patterning. Together, this provides significant new evidence for early intrinsic programming in the human brain. This analysis will cover 6 sections. We'll begin with the core spatial problem in the field, then describe the spatially resolved atlas that was created to solve it. From there, we'll examine the findings on early lamination and dynamic gene programs. This is followed by the paper's central discovery of 2 distinct modes of arealization. And finally, we'll discuss the broader functional and conceptual implications of this work.
Section 1. A fundamental spatial problem, and why spatial context is essential.
Over the last decade, developmental neurobiology has been truly transformed by single-cell genomics. The most common method, dissociative single-cell RNA sequencing, involves breaking a tissue sample down into a suspension of individual cells to profile the respective transcriptomes. Now, while this approach has been instrumental in generating a molecular parts list for the brain, identifying hundreds of cell types, its core methodological step, the dissociation itself, imposes a critical limitation. This illustrates a central trade-off in cellular genomics. Dissociated methods offer incredibly high-throughput cell typing, but it comes at the cost of spatial context. All information about a cell's original location and its neighbors is lost. Conversely, spatially resolved methods preserve that precious tissue architecture, but they've historically offered lower throughput and lower molecular resolution. The technical challenge this paper addresses is how to bridge this gap between high-resolution molecular profiling and high-resolution spatial mapping. To really understand brain development, it's not enough to know which cells are present, we have to know their precise organization. This loss of spatial information is a critical barrier to resolving long-standing debates in developmental neuroscience. For instance, the mechanisms of cortical arealization. The protomap hypothesis posits that the layout of cortical areas is intrinsically programmed. Here, progenitor cells in the ventricular zone are patterned by morphogen gradients, and this spatial information is then inherited by the neurons they generate. In contrast, the protocortex hypothesis suggests the cortex is initially an unpatterned structure, with area-specific identities induced later by external inputs, mainly from the thalamus. To adjudicate between these models, we need precise spatial mapping of molecularly defined cell types during early development, and this is exactly the information that's lost in dissociative studies. This fundamental problem demands a methodological solution capable of generating a high-resolution, spatially intact atlas of the developing human brain.
Section 2. A spatially resolved atlas. The methodological solution.
To construct their atlas, the authors employed a multimodal pipeline. First, they used MERFISH, or multiplexed error-robust fluorescence in situ hybridization, to image hundreds of selected gene transcripts directly within intact tissue slices, which preserves their spatial coordinates. Second, to overcome the challenge of the extreme cell density and fetal tissue, which makes traditional segmentation algorithms ineffective, they developed a custom deep learning model to accurately delineate individual cell nuclei. Third, they integrated this high-plex spatial MERFISH data with deep transcriptomic data from single-nucleus RNA sequencing. This multimodal integration allowed them to impute the expression of thousands of additional genes onto the spatially mapped cells. Finally, these integrated data sets from multiple samples were assembled into a comprehensive atlas. We're looking at a schematic from Figure 1A of the paper, which details the study's sampling strategy. What is being shown are 2 views of the human fetal cortex, with colors indicating the 8 distinct cortical areas that were sampled, spanning the anterior, posterior, and dorsal ventral axes. You can read this by noting the comprehensive spatial sampling, which was repeated across 7 different developmental time points, from gestational week 15 to 34. The key comparison here is the combination of both spatial and temporal sampling. The authors conclude that this design is sufficient to capture developmental dynamics across the entire cortical sheet during mid-gestation. This panel advances the paper's argument by establishing the robust and comprehensive nature of the dataset from which all subsequent conclusions will be drawn. The resulting atlas is massive, comprising over 18 million individual cells, each with an assigned cell type and precise spatial coordinates. This scale provides the statistical power and cellular resolution that's required to detect subtle patterns of spatial organization that would otherwise be undetectable in smaller datasets. Now, before analyzing spatial patterns, it's necessary to validate the cell type classification. This is a UMAP plot from Figure 1C. What's being shown is a dimensionality reduction of the transcriptomic data from all the cells in the atlas, where each point represents a single cell. You can read the panel by observing the proximity of points. Cells with similar gene expression profiles cluster together. The key pattern here in this abstract molecular space is the formation of distinct clusters that correspond to the major known cell classes: excitatory neurons, inhibitory neurons, and various glial populations. The authors conclude that the clear separation of these clusters demonstrates successful and accurate cell typing. This panel is critical because it validates the molecular identities of the cells that will then be mapped back into their physical spatial locations for the core analyses of the paper. With a validated, spatially resolved atlas of cell types now in hand, the authors were positioned to investigate the spatial organization of the developing cortex. Their first question concerned the vertical axis, the formation of cortical layers. This brings us to Section 3, early molecular lamination. This panel from Figure 2C shows a diponuclear stain of the cortical plate at gestational week 22. What's being shown is a dense field of cell nuclei, representing the classical histological view of this developmental stage. To read this, you observe the distribution of nuclei from the top, which is the pile surface, to the bottom, the white matter. The key spatial pattern here is the absence of clear organization. The nuclei appear as a largely uniform and undifferentiated field. The established conclusion from images like this is that the 6 distinct cytoarchitectural layers are simply not yet morphologically apparent. This panel is important because it establishes the baseline classical view that the authors are about to challenge with their molecular data. In stark contrast, this panel, also from Figure 2C, shows MERFISH data from the exact same developmental stage. What's being shown here is the spatial distribution of distinct excitatory neuron subtypes, where each point is a single neuron colored by its molecular identity. You read the panel by observing the vertical organization of these colors, from the pile surface down to the white matter. The critical spatial pattern is the clear stratification of colors into 6 distinct bands. This reveals a highly organized, 6-layered structure that is defined entirely by the molecular identity of the neurons. The authors conclude that this molecular lamination is established months before classical cytoarchitectural layers become visible. This panel is absolutely central to the paper's argument because it provides direct evidence of a cryptic spatial organization that exists far earlier than previously thought, an organization that's only visible with spatially resolved molecular methods. The authors state their conclusion directly. This early molecular specification likely serves as a foundational scaffold for all subsequent circuit development. Now, the discovery of this cryptic organization raises a further question. Are the gene programs defining these early layers static, or do they evolve over developmental time and across cortical space? Section 4, Dynamic Laminar Gene Programs, an Evolving Molecular Identity. It turns out the molecular definitions of these layers are not static at all. This heat map from Figure 3D illustrates this dynamism. What's being shown are genes that define specific layers, with their expression plotted across different developmental time points and cortical areas. To read the panel, you follow the rows, which represent genes, across the columns, which represent different spatiotemporal contexts. The key pattern is that the genes with high expression, which are indicated by color intensity, change depending on both the age and the cortical area. For example, the set of genes defining layer 4 in the frontal cortex at week 20 is distinct from the set defining layer 4 in the occipital cortex at week 30. From this, the authors conclude that the gene expression programs specifying laminar identity are highly dynamic, varying across both development and cortical geography. Crucially, these fetal laminar gene programs are not simply immature versions of the gene programs that define layers in the adult brain. Instead, they represent unique, transient developmental states. These are molecular programs that are tailored for specific developmental tasks, like migration and initial axon guidance, which are later replaced by programs responsible for mature circuit function. This finding argues against a simple linear model of maturation and points instead to a much more complex, stage-specific regulation of cellular identity. This dynamic molecular identity of the vertical layers provides a new framework for investigating the horizontal organization of the cortex into distinct areas. So now we pivot from the vertical axis of lamination to the horizontal axis of arealization. This really is the conceptual heart of the paper. Section 5. 2 modes of arealization, gradients and a sharp divide. Based on previous work, particularly in rodents, the dominant model for cortical arealization involves smooth gradients of gene expression. And indeed, the authors find strong evidence for this first mode of arealization in their human dataset. When they examined the distribution of neuronal subtypes across most cortical regions, they observed continuous, gradual transitions. For example, the molecular identity of neurons in the frontal cortex changed smoothly as one moved posteriorly towards the parietal cortex. This graded pattern fits well with the idea that broad positional information is established by morphogen gradients, which is consistent with the proto-map hypothesis. However, when they examined the occipital cortex, which contains the visual areas, they revealed a second discrete mode of arealization that contrasted sharply with the graded transitions they observed everywhere else. This composite from Figure 4 presents the central evidence for this discrete mode of arealization. On the left is a DAPI stain of the occipital cortex at gestational week 20. What's being shown is a morphologically uniform field of nuclei with no visible anatomical boundary. On the right is MERFISH data from the same region. Here, what's being shown is the spatial distribution of excitatory neuron subtypes, colored by their identity. V1-enriched subtypes are in warm colors, while those outside V1 are in cool colors. To read the panel, you compare the 2 images and observe the distribution of colors in the MERFISH data. The critical spatial pattern is the abrupt transition from warm-colored V1-specific neurons on the left to cool-colored neurons on the right. This forms a sharp molecular border where no morphological border exists. The authors conclude that a precise molecular boundary between the primary visual cortex, V1, and adjacent areas is established by gestational week 20. This panel is the conceptual core of the paper, as it reveals a sharp, discrete mode of arealization not predicted by gradient-based models and provides very strong evidence for the intrinsic patterning of a functional boundary. The existence of this early and incredibly precise molecular boundary between V1 and other areas really necessitates an investigation into its functional significance. Specifically, does it reflect an early commitment to the functional specialization of these neurons?
Section 6. Functional and conceptual implications of this early patterning.
To investigate the functional implications, the authors analyzed genes that were specifically upregulated in V1-destined layer 4 neurons, the primary recipients of thalamic input. They found a significant enrichment for genes involved in synaptogenesis. This suggests that even at this early stage, V1 neurons are already expressing the molecular machinery to form the circuits needed for visual processing. This finding supports 3 main conclusions. First, cortical areas began preparing for their function far earlier than we previously knew. Second, the cortex employs at least 2 different arealization modes, gradual gradients for broad patterning and sharp boundaries for specific functional areas. And third, the existence of this intrinsic sharp molecular boundary provides strong support for the proto-map hypothesis in humans, indicating that a detailed cortical blueprint exists prior to any significant thalamic input. In conclusion, the authors summarize their contribution with this statement. Collectively, our findings underscore the crucial role of spatial relationships in determining the molecular specification of cortical layers and areas. This work provides a powerful demonstration that to understand how a complex tissue like the brain is built, we cannot simply study its constituent cells in isolation. It's the precise spatial arrangement of these cells, their organization into layers and areas, that defines the structure and ultimately the function of the cerebral cortex. This atlas provides a foundational resource for exploring the developmental origins of human cognition and neurological disease, leaving us with a deeper appreciation for the intricate and genetically encoded program that constructs the human brain.